Excellent. Okay, well, welcome everyone. My name's Miriam.
And I'm Jeremy.
And we're both scientists at the National Heart and Lung Institute, and we're going to talk to you about your lungs, your linings of your lungs, and the bugs in your lungs today. So have any of you heard of the Human Genome Project? One person. This was a project that started in 1990 and finished in the year 2003, so 13 years. And as a result of it, the whole sequence of the human genome became known. And so we now know that we have 20,500 genes. These genes make the molecules that are the building blocks of our cells. So since then, in 2008, a new project started known as the Human Microbiome Project. And this is to characterize all the bugs, so this is bacteria, viruses and fungi that live on you and in you, and that are associated with you when you're healthy and also you when you're diseased. So if I tell you that the number of cells in your body are 10 to the power of 13, so this is 10 with 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So that's the number of cells in your body. Now, how much do you think of you is microbial, so bacteria, non-human? Do you have any idea? 95.5%. Not quite actually, but you're quite close.
I guess, yeah.
So 75% of you is not human and 25% of you is human. Now you shouldn't be alarmed about this because these bugs, the microbes, are very important for your health and they're very small. So medical teaching was that the lungs...
Sorry, 75% of the cells in your body
are non-human in origin. So the bugs that live on you and in you. So the ratio is 3 to 1. For every human cell, you have 3 microbial cells. But it's fine because they're very important for keeping you healthy and well. So medical teaching was that the lungs were sterile, but do you think that could be true? Think, how much air do you breathe in each day, in and out? So this is to represent the air. And in the adult, how many of these do you think you breathe in and out each day? 10,000 of these. So 10,000 litres a day when you're you're a young boy at the moment, but when you are a young man and a young adult, you will breathe in and out 10,000 of these. And obviously, air is very important. It contains the oxygen that we need in our bodies, but also it has foreign bodies and germs that we breathe in and out. And that's why the structure of our lungs is very important. So I like to show this as an example of the structure of your lungs, the internal structure. Now, if I turn it up this way, it will hopefully make more sense. So this is where your mouth, and you breathe in into your windpipe, and then it branches. And as you go further into the depths of your lung, you get many more branches until right at the end you'd have the leaves. And that's where you get the oxygen into your blood. Now, the lungs have a very special lining. And if we took out the lining, what area do you think it would cover if we laid it out? Would it be, say, as large as this board, do you think? That is correct. If you took it out, the surface area would be the same as a lawn tennis court, and that's all contained within you. Now, the cells lining the lungs are very important. They're called epithelial cells, and some of them have lovely hairs, and these are important for wafting out the germs and the things you don't want in up into your mouth, where then you can swallow them. Also, there are special cells that produce mucus, and this is a bit like the car engine oil to keep everything nice and lubricated and working well. Now, we did a study at the National Heart and Lung Institute because we felt that the lungs were not sterile. And we did the study using new techniques to actually look at the DNA present, the DNA of the bacteria. And you can learn about that in a later demo called Who's Living in Your Lungs. Now, obviously, it would be lovely to be able to study the baby when it's in mummy's tummy, but clearly we can't really look at the baby's lungs at that point. So we did a study of babies that had been born, and we looked at them early on in life at 7 months and 12 months. and we looked at what their lungs were doing. And it was a little bit like seeding a wild flower meadow. At the start, there were just a couple of little bugs present, or in this case, flowers. And then much later on, we saw more and more of the flower meadow emerging until we have a beautiful flower meadow. So this is the equivalent of all the bacteria in your lungs. You have a lovely little community. You do have some odd, not so attractive ones. And so what we've discovered is if, for example, you get a cold, suddenly the beautiful flower meadow can be dominated by one rather ugly, showy flower. So we've been doing all these studies of different diseases like asthma, COPD, chronic obstructive pulmonary disease, and we now know there are different communities of bacteria that are important in causing these diseases. And you can learn more about them at the microbial pick and mix. But having identified these, the next thing is we want to know how do we respond to the bugs that are present in our lungs. And this is where Jeremy comes in.
So, as Miriam said, the cells that line your airways are all the way in here, very hard to get. So does anybody have any idea how we could get cells out of the body to study them? Yeah?
Or like if you went to like a hospital or something, they put like a tube down your throat
Yeah.
and they'd use like suction.
Close, yeah. They can actually use a brush. Spot on, well done. When some people go in for an operation and the anaesthetist knocks them out and they're asleep and they have the tube that helps them breathe, we're able to put a brush down their airway and brush the cells. Now I'm going to do that to Miriam now, okay? So if you just watch. I'm not really. That's far too big. But this is actually the brush that we do use. So you can see it's very, very long because it's quite a long way to get all the way down into your lungs. But if I push this, you'll see out of the end, and Miriam's going to come around and show you it if I hold it steady, a tiny little brush. So Miriam's going to bring that round and show you. And that is a miniature version of this. And what that allows us to do is get lots of cells, about 25,000, which sounds like a lot, doesn't it? But actually, in order for me to do my experiments, I need about 6 million. So whenever I get the brush from the doctor, I put the cells into a tiny little flask, and they grow on the bottom of the flask in something called media. So this is what the cells eat to help them grow. So when you guys go home and eat your dinner and that helps you grow, this is what the cells are eating. It doesn't look very nice, does it? Once the cells have grown in this little flask, I take them out and I put them into this big flask. And once they've covered this big flask, that gives me the 6 million cells that I need to study them. But that's not them finished. They have to do one more move from this flask into this plate. And if you can see in the plate, there are about 12 little wells that if I lift them out are suspended inside a bigger well. See that? And that's where the cells grow. And over 28 days, over about a month, the cells form what's known as well-differentiated epithelial layer. That sounds a bit complicated, so I'll explain it. Here are the cells that line your airways that produce the mucus and the cilia. And over 28 days, these cells form and they become very specialized. As well as that, they form a barrier. So if you look at your skin at the minute, your skin is a barrier, isn't it? So any bugs or any pollutants that are in the air can't get past your skin. Your skin's there to protect you from all that. It's exactly the same in the lungs. Your lungs form this barrier, and the yellow dots are all the nuclei of all the different cells in your lungs, and the green lines are those tight junctions that help form the barrier, and that's what protects your lungs. As well as that, we've got special cells in the lungs. So these are some of the cells that were taken out of somebody's airway, and then I grew them in this little dish here. And what you can see is that there's about 3 or 4 cell layers thick, and they're cells called goblet cells. These produce mucus. So you see the way you just coughed there? If you were to cough up mucus, these are the little cells that produce the mucus. Also, there are these little hair cells called cilia, and it's just like on your head. If you imagine your hair was constantly moving like that, that moves the mucus up all the way up, and you either cough it out or you swallow it, which is a bit gross, but that's how your airways protect themselves. So what we can do is, having grown these cells, we can put some of the bugs that Miriam was talking about onto the cells, and we can study how all the genes of the airway are affected by these particular bugs. And Miriam's going to explain a little bit more about that
And
now.
so why that's important is, as I highlighted, we have 20,500 genes that make up the special molecules that are the building blocks. But obviously each of the microbes, each of the bacteria has its set of genes. And so they have over 1 million plus genes that we're interacting with. So the way we can look at what we're doing is using this special array that allows us to measure all 20,500 genes simultaneously. So one of these is one array. So we'd look at one person's sample from Jeremy's experiment and be able to measure all 20,500. So we can do 24 individuals at once. But in fact, if this plate was full, we could do 96 individuals. But this then would cost us £10,000 just for this small piece of item to make all those measurements. So it's expensive, but by doing this, we can learn how we're responding to the bacteria present, we can understand which genes are important, and we can then develop new therapies and targeted medicine to help us in getting the community balance right to the healthy state.